I'm Charlene Walker from the lab of Charles Kimmel at the University of Oregon, the Institute of Neuroscience.
And I'm Gregory Walsh. I work in the lab of Cecilia Mowens in the Division of Basic Sciences at the Fred Hutchinson Cancer Research Center.
Today we will show you a procedure for making gynogenetic diploid zebrafish by the early pressure method. This method uses hydrostatic pressure to depolymerize microtubules in an egg fertilized with UV irradiated sperm. Done with the right timing, this prevents the expulsion of the second polar body, generating a diploid embryo, whose homologous chromosomes are the two products of meiosis I. This technique was worked out in the early 1970s when I worked in the lab of George Streisinger at the University of Oregon.
In our lab, we use this procedure in 2-generation forward genetic screens. The gametes of F1 mutagenized fish can be screened directly for recessive mutations that cause developmental phenotypes in homozygous gynogenetic diploid offspring. Since zebrafish exhibit high genetic interference, we also use this method to map mutations to chromosomes and to estimate how far these mutations lie from the centromere.
So let's get started.
During the late afternoon the day before the experiment, prepare for sperm collection by separating males and females to be squeezed. Old males can be used since they will not be contributing any genetic material. Females should look large and fat in the belly, while males should look yellow and spry. Hold the males and females overnight in separate holding tanks where they can be easily accessed in the morning. On the morning of the experiment, prepare Hank's solution and add sodium bicarbonate. Bring to the fish facility in an ice bucket the Hank's solution, tricane, a timer, and tubes for collecting sperm. Finally, once in the fish facility, dilute the tricane in fish water. This will be used to anesthetize the fish in the next step. To begin sperm collection, prepare a clear vial of Hank's solution on ice. Chill enough Hanks for about 50 microliters per male to be squeezed. Anesthetize 1 or 2 males at a time by immersion in tricane. They can be lifted out of the anesthetic with a plastic spoon. Rinse the males in fish water and blot damp dry on a paper towel. This is very critical because water activates sperm. Once they are dry, mount the fish in a foam holder under a stereo microscope at low magnification with epi-illumination. Separate the pelvic fins with forceps and position a microcapillary at the opening of the cloaca. To begin collecting sperm, stroke the sides of the fish gently but firmly with smooth millipore forceps. As the milky sperm comes out of the genital pore, collect it in the microcapillary using gentle suction. Pool the sperm from several males in ice-cold Hank's saline. Sperm from 5 to 10 males is adequate for fertilizing eggs from 20 to 30 females. Sperm kept in cold Hanks will continue to fertilize eggs efficiently for several hours. Collect enough sperm to make a cloudy suspension. To UV inactivate sperm, start by filling the bottom of a glass petri dish with ice. Put a watch glass on top of the ice in the dish. Next, use a pipette to transfer the sperm from the test tube onto the watch glass. Be sure to get as much of the sperm into the pipette as possible without taking up any bubbles of air. It is critical to avoid bubbling the sperm. Expel the sperm in a thin, even layer on the watch glass and discard the pipette. After the sperm has been transferred, place the petri dish into a UV crosslinker. Turn on the crosslinker set on energy to deliver 900 joules. After cross-linking, use a clean pipette to remove the sperm from the watch glass and transfer it to an Eppendorf tube marked UV sperm and place on ice. To begin egg collection, first anesthetize 2 females at a time in tricane. It is critical not to keep the fish in tricane too long because prolonged anesthesia can kill the fish. Next, rinse the fish in fish water and blot damp dry on a paper towel. This is critical because excess water will swell the eggs and prevent fertilization. Place the female in a 35 mm plastic petri dish. With damp but not wet fingers, press gently but firmly on the belly. If she is ready to lay eggs, they will come out easily. Gather the eggs with a spatula and return the female to water. Good eggs are a yellowish, translucent color, whereas eggs that have remained in the female too long are white and watery. To ensure getting good eggs, collect them during the first 2 hours after the lights come on in the fish facility. Keep the eggs covered to prevent drying out. After collecting the eggs, add 30 to 50 microliters of the UV sperm suspension to the eggs and stir gently with a pipette tip. Immediately add about 1 milliliter of fish water and start the timer. Eggs left to develop at this point will be haploids. The following steps must be accomplished within 90 seconds of fertilization. Start the timer at the moment that eggs are activated by the addition of water. The exact timing of the metaphase of meiosis I is temperature dependent. The timing of pressure on and off is optimized for 28.5 degrees Celsius. After starting the timer, wait a few moments. Then add more water and swirl the eggs to the center of the dish. Using a cut-off and polished Pasteur pipette, transfer the fertilized eggs to the pressure vials. If needed, add more egg water to the vials so they are almost overflowing, leaving a bead of water at the lip of the vials. Next, place a rubber top over the bead of water and secure the plastic lid over the rubber. Place the vials upside down in the pressure cylinder, adding more egg water to refill the cylinder. Put the top on the cylinder securely. Once closed, place the cylinder in the press with the plunger up and apply pressure of 8,000 pounds per square inch. Leave the cylinder in the press until 6 minutes after the time of activation, for a total time of 4.5 minutes of pressure. Six minutes after activation, remove the cylinder from the press. Remove the vials from the cylinder and carefully dry them to prevent cooling of the embryos. Reset the cylinder and refill it with egg water. Prepare the next vials by partially filling them with egg water as needed. Repeat these steps for all the batches of eggs. The cylinder will hold up to 2 vials at a time, so you may want to delay fertilization of eggs 1 to 2 minutes to see if other females already in the tricane give eggs. This will help speed things up by treating more batches at one time. Also, if you squeeze fish while the press is being used, the eggs may be too dry to be viable by the time the press can be used again. 24 hours after fertilization, a haploid zebrafish embryo will have a short, stocky body, a short tail, and less clearly formed brain, yet they will all have the same body parts such as somites, notochord, eyes, and ears as diploids. Haploids and diploids all exhibit uninhibited flexing motions around 24 hours post-fertilization. EP diploid siblings have long tails, nicely sculptured brains, and should be about 60 to 70% viable. These look very much like the untreated diploid controls. Usually, a batch of haploid embryos will have a mixture of good-looking embryos, dead embryos, and everything in between. The proportion of good-looking embryos to defective ones reflects the deleterious genes harbored in the female genome. Mutations are relatively easy to identify even with this background, because there will be several individuals, up to 50%, with the same characteristic phenotype. It is important to make sure that the embryos produced by this method are true gynogenetic diploids. As controls, generate a clutch of normal diploid eggs by keeping aside some of the sperm without UV inactivation, and a clutch of haploid embryos by keeping aside a clutch of eggs fertilized with UV sperm without going through the EP procedure. Haploids are not viable after 2 to 3 days. EP diploids should look like normal diploids, although some irregular EP monsters may occur. Clutches of EP diploids should be largely viable, around 70 to 90%. The presence of haploids in the EP diploid clutch suggests a failure in the EP process, for example, the press failing to hold pressure. The presence of diploids in the haploid clutch suggests incomplete sperm inactivation, for example, a failure in the crosslinker.
We've just shown you how easy it is to do in vitro fertilization with zebrafish, and how to make gynogenetically derived diploid zebrafish embryos by inhibiting the second meiotic division.
When doing this procedure, it's important to remember that the timing of the early pressure is precise and that the press holds its pressure at 8,000 pounds per square inch throughout the treatment. Be sure to have your press calibrated.
So that's it. Thanks for watching, and good luck on your experiments.